The overall goal of the following experiment is to monitor the accumulation of the disease-associated α-synuclein in a transgenic mouse model of Parkinson's disease, using a quantitative ELISA approach. This is accomplished by first allowing uninoculated mice to age normally, or by inoculating mice intracerebrally with a brain extract from sick M83. After the mice develop clinical disease and are sacrificed, brain regions from the mice are dissected. Next, homogenates from each brain region are prepared in high salt buffer and ELISA is performed on the homogenates in order to quantify the disease-associated α-synuclein in each brain region. The ELISA results show that the disease-associated form of the α-synuclein protein is mainly accumulating in the caudal brain regions, including the mesencephalon and the brainstem, as well as in the spinal cords of the mice.
The main advantage of this ELISA technique, compared to previously available methods like Western blot or immunohistochemistry, is that it provides rapid and reliable quantification of the disease-associated alpha-synuclein in this experimental model.
This method can thus help monitor the accumulation and the propagation of this protein in the central nervous system of these M83 mice. That suggests the involvement of a prion-like mechanism up to the development of the clinical signs. Further developments of this method could be considered in the future for the study of various biological samples such as the cerebrospinal fluid or the blood that possibly also contain altered alpha-synuclein forms. Demonstrating